Good afternoon, everyone. I think we have a critical mass of people who joined and for us to start. I'm very happy to introduce today Jaan. Jaan is associate professor from the University of Tartu in Estonia. Jaan is working on neural correlates of consciousness, has been working on many different levels, from cellular level, system level, and also use of artificial intelligence model. And we actually got several requests from people here at the center to invite Jaan for a talk in the seminar. So I'm very excited to have him today with us. And Jaan, the stage is all yours.
Thank you very much. And I'm very delighted to be here, and I'm very happy about the invitation because I've been reading many papers from the people in your faculty, and these are really some groundbreaking papers. So I'm happy to present some of my work. And it is about consciousness. Now, I've been studying consciousness since my undergraduate studies, so 17 years. And the fundamental aspect about consciousness is that if you ask someone, what is consciousness or how is it generated in the brain, then the answer has to be, we do not know. However, then there are these theories of consciousness that basically say, yes, we do. We know what consciousness is. And these are the four most dominant theories of consciousness. Sorry for the acronyms, I actually won't explain them. Some know them, others not. The point is that these theories take up a lot of space when it comes to studying consciousness, in the sense that roughly 90% of research is dedicated to studying these theories. And some of you might have heard it, some of you might have seen it, that in the recent years, the Templeton World Foundation actually invested more than 10 million euro into studying these theories. And of course, it's very nice that we test these theories against each other. It's important for science, I agree. But that also got me thinking, is this really the best investment when it comes to studying consciousness? Or is there something else we could do? And the view I'm advocating today is that instead of really pitting these theories against each other, we could be at least also developing new theories of consciousness. And I try to advocate for this point and show you one example. So, of course, when we look at these theories, they are prominent theories. It's very important to study them. And I would, of course, also agree that it's very important to test these theories against each other. However, if you look at them, then all of these theories were established more than 20 years ago. And it's not necessarily bad to be an old theory. However, in our research, when it comes to neuroscience and cognitive science and computational neuroscience, then, there have been several revolutions since then, right? And I only mention two of them, which probably you both know, the first being simply the neuroscience revolution, right? Back then, when these theories were established, we did not have optogenetics. We did not have all these tools for manipulating the circuits of the brain. And I won't be going into this image here because I think all of you know that, with optogenetics, you can manipulate specific neurons. You can do this to different types of neurons. You can manipulate different parts of neurons, different connections. And eventually we want to actually take that into account. So just to bring one example, back then, 20 years ago, people thought that, what the neuron does is something like depicted here, like that it integrates the inputs sums it, and then when it crosses a threshold, then it sends out a spike. But now we know, and also thanks to work done in your institute, we know that neurons are much more complex, right? Here's just one image that we used in our paper to show that, I mean, this is a single pyramidal neuron, layer 5 pyramidal neuron. We can say that it has different zones, different parts. There are different types of spikes. There is a specific connection coming in from higher order thalamus, etc. All of these details were still, or many of these details were still largely unknown 20 years ago. And there are several of these kinds of revolutions. I mean, one revolution that I won't mention today, but that is highly relevant, is also the revolution in artificial intelligence. But the second revolution I want to mention is a revolution in how we think about the effect of top-down processes on conscious experience. And you all have seen these kinds of effects, and indeed, most of them were known already 20 years ago, 30 years ago, even more. However, these kinds of top-down effects were not really important parts of a theory of how we think about what the cortex does or how processing happens. And of course, one paper that came out of your university laid the groundwork for that in 2002, and has been the pioneering paper for changing this view that is currently dominating, namely that all of our conscious experiences are affected by prior knowledge, that this is something very pervasive. Whenever we perceive something, it is modulated to a larger or lesser degree by prior knowledge. Just to bring one example from our research, here you have an everyday object embedded in noise. So we have rendered the everyday object with machine vision tools into dots, and then added noise. And your task is to figure out what kind of an object is embedded here. Some of you will see it, many of you won't. But when I now simply tell you that look for the classes, then I guess, at least based on our experiments, many people see that, oh, indeed, here are classes. And the question is, what exactly happened in your brain? What exactly happened in your conscious experience after giving this kind of a top-down cue? And this is, of course, just one example of a top-down effect, but it's an example that brings us to the question of how can actually these theories that I just showed you explain these pervasive effects of prior knowledge and top-down knowledge. And sure, actually, all of these theories somehow can explain that. The global neural workspace theory now has been using the predictive coding view to actually explain some of these effects. But the point is that they have to make extra assumptions to do that, because this pervasive nature of prior knowledge was not kind of embedded in these theories. And then the question becomes, should we try to patch together these old theories of consciousness, or rather should we try to build new ones that take these revolutions into account? And, you know, in the end, the question is about the balance. Because these large sums of money went now into testing old theories of consciousness, then I'm here to say that the balance is tilted in the wrong way. And at this moment here, where we have so much new knowledge about the brain, we should be trying to develop new theories. And that's what I will give you an example of. So I will give you an example of one new theory of consciousness that we have proposed. And just to be very clear here, I'm not saying that this is now the best theory and that all the others are history. But what I am saying is, look, here is an example of a new theory of consciousness that is based on these revolutions, that takes these kinds of new data from the brain and also from our conscious experience into account. So let's get into it. The theory is largely based on what the layer 5 pyramidal cells do. And some of you know this very well, but for the others, layer 5 pyramidal cells have their cell bodies in layer 5. But what is so exciting about them is that their dendrites span all the way to layer 1 of the cortex, so all the way to the surface of the cortex, where they expand and they collect a lot of input. And similarly, the output of these neurons is important, because indeed there are other cell types like layer 6 thalamocortical cells that connect to subcortical structures. But the layer 5 cells, they have much more extensive projections to the striatum and to the non-specific nuclei of the thalamus. And layer 5 pyramidal cells even send projections to the brainstem. So, I mean, it makes sense to say that these layer 5 pyramidal cells are the key output structures of the cortex. So when you would deactivate all these layer 5 pyramidal cells, then the cortex can do all of its computations, but it wouldn't really matter. There's no effect, almost no effect on the behavior. So that's why these cells are interesting, and why historically they have been considered to be a key players within a theory of consciousness. However, the last 20 and a bit more years have shown that, you know, they have some very exciting properties. And for simplicity, we can say that, you know, we can look at these kinds of neurons as of having two key compartments. One is the basal compartment here around the cell bodies, which we could, which is here on the bottom of the figure. And the other is the apical compartment around the layer 1, layer 2, and the duct dendrites. And here, people have suggested that it's not simply that they are two anatomically separate compartments, but that this is this has a functional meaning. And my postdoc supervisor, Matthew Lackum in Berlin, was one of the earliest and probably the loudest to say that, look, it's not simply an anatomical distinction, but these two parts of these pyramidal cells, they get fundamentally different input, where the basal compartment receives something like a bottom-up information or a feature information, and the apical compartment receives top-down information, or as Matthew likes to say, context. So this is important. It's not simply an anatomical distinction, but it's a functional distinction. And indeed, when we look into the brain, then we see that, you know, there are a lot of areas projecting now. Now we see slices of the primary somatosensory cortex in mice, and we see then areas projecting to layer 1, for example. We see this is the secondary somatosensory cortex, but also auditory cortex, perirhinal cortex, motor cortex, or in this case, frontal cortex, and then all these thalamic areas that they do send some collaterals, sometimes more, sometimes less, to layer 5, to other layers, but they have this very distinct projection to layer 1. And Matthew has said for a long time that this is now no coincidence, that this is one of the most important organizational principles of when we want to understand the cortex. And then the experiments which some of you know and which made Matthew famous were these experiments where he took these layer 5 pyramidal cells and he did these really complex experiments where he injected current into these different compartments of the pyramidal cell and measured what happens if you activate them simultaneously. And in short, here what you can see on panel D is that when you activate these two compartments simultaneously, then you have something like a superactivity. It's not simply a sum of the activation of the apical compartment and basal compartment. It's something qualitatively different. And then the first very important conclusion that Matthew made already back then was that, there is a cellular mechanism in each of these layer 5 pyramidal cells that signals a match, that a match has been made between these two streams. And remember, these are not simply some two streams, but they are functionally different two streams. And that each of these layer 5 pyramidal cells has an intrinsic mechanism to combine these two streams. So let me get to a very important point that, what are these two streams? And as said, Matthew liked to use the terms context and feature, but the point we want to make is that the names actually do not matter that much. You could say that, what the apical compartment receives are predictions. You could say that it's feedback. You could say that it's the generative model or the internal model. And you could call them higher order representations that activate the apical compartment. And vice versa, when you go to the basal compartment, you could say that this is input from the external world, that this is feed forward, these are prediction errors. These names do not matter. The point is that anatomically, in the brain, when we look at these two compartments, they receive functionally distinct input so that the apical compartment receives something that is, we can say, is internal or top-down. And the basal compartment receives something that we can say is bottom-up or external. So we can call them different names, but biology matters here. Biology says that they are distinct streams with distinct functions. Now, you could ask, well, what does it all have to do with consciousness? And I think for many years, Matthew has had this idea that this is related to consciousness, but he also couldn't really pinpoint it. But then when I went to Matthew's lab, then there was a postdoc, Mototaka Suzuki, who showed me this amazing result that he just had obtained. So what he did, he used optogenetics to activate the apical compartment. And when he did that, so in vivo, in awake animals, then he saw that, when you activate the apical compartments, you can measure the spiking response in the pyramidal neurons in the basal compartments. Okay, but the really exciting discovery was that this did not happen when he anesthetized the animals. So when he anesthetized animals, then he activated the apical compartment the same way, but there was silence here. There was nothing evoked by the stimulation of the apical compartment, as if this propagation from the apical compartment to the basal compartment was cut off. And this is then the exciting part, that here we have a comparison between a conscious state and an unconscious state. He did then the proper controls by trying out different anesthetics, by doing this experiment in different brain areas. And he confirmed that, that at least based on his experiments that were published in Cell, the the difference between these two states is really this state of consciousness, right? Now, and again, that there's something that happens in the state of when the animal is conscious, that there's this propagation from the apical compartment to the basal compartment that does not happen under anesthesia. And this mechanism is a bit more complex because it's controlled by higher order thalamus. So he could show that higher order thalamus controls this kind of a coupling. I think I won't go into that today. If you want, if you're not so a hardcore neuroscientist, but you want to understand it, then in our Trends in Cognitive Sciences paper, we explain that mechanism. And if you want, then you can also go to this, to the paper by Mototaka and Matthew. And maybe one point I want to make here is that, you see this discovery was published in 2020. So 20 years ago, we had no idea like that. We had no idea that this is possible, that there is a gate somewhere between the two parts of a single neuron, and that this gate somehow is related to consciousness. But now we know. So when I went into the lab of Matthew and I started to work on this, then I said, well, wow, we have to actually put this together to a theory of consciousness. And this we did. So in very short, before I go on to explain this theory, the point is that what this finding showed is that in unconscious states, you have this decoupling between the two parts of the pyramidal cells. And in conscious states, they are coupled. So from this perspective, we have a cellular mechanism that explains why conscious processing is affected by top-down and prior knowledge. It is inherent to this theory because what separates conscious from unconscious states is this functional separation of the top-down information and the bottom-up information, or if you want in even more generic terms, unconsciousness is when there's a decoupling between the internal and the external. And that is the basis of the theory, and now it's a good point to see if there are any questions.
Yeah, there's a question from the audience, from Ada Levi. She asked regarding Suzuki's results, was the stimulation also attempted during sleep?
Yeah, excellent question. Excellent question. And of course, I mean, this is, for me, this is the beauty of this theory, right? That we can immediately, and basically everyone can see why this question makes sense. Does this also happen in non-REM sleep? And indeed, this is what we would predict. So this is a very hard prediction of the theory, that you would see exactly this kind of a decoupling in NREM sleep. But until today, this experiment has not been done, partly because Mototaka himself now went to Amsterdam and has been not able to get his experiments going. But this is an experiment that we want to do. And of course, we would be delighted if somebody else actually does it. Yeah, thanks.
I have another question.
Yeah.
Go ahead.
Yeah. Sorry, who wants to?
Just ask at the same time. Ask at the same time, and I try to answer both. Go ahead.
Go, Mark.
I don't know if you have read the work of Marshall DeVore. It's the lab that I work in, but he has one critical paper in an experiment where what he does is he injects GABAergic anesthetics into a brainstem locus, a very specific locus in the brainstem, and he gets unconsciousness. But when he lesions this tiny area of the brainstem, he puts systemic anesthetics in the same amount that causes consciousness, in the same concentration, and now unconsciousness, and now it doesn't. So you're flooding all the cortex with the same amount of anesthesia, but now you're not getting unconsciousness. And you would think that in this theory, it will prevent all the pyramidal neurons to this top-down effect that you mentioned.
Yeah, I wish I could answer better, but I unfortunately haven't read the paper. Please send it to me, and then I will be
sure.
in a better position. Sorry,
Sure,
sorry, really.
I would love to talk about this with you later.
Yeah. Thanks anyway.
And Jaan, I have a short question. Do you think it's just a lowering of the baseline level of activity, or are you suggesting there's a different mechanism there?
No. I mean, we really think, and the experiment by Suzuki and Laukom also suggests that it's not simply a lowering of activity, but fundamentally that this is this change in the coupling. What he does, so Mototako, what he did, he changed the level of activity, so you can change the intensity of the stimulation. And essentially, it's not that, at higher intensities, the same mechanism kicks in unconscious state also. No, it's nothing in unconscious states, and you get it in conscious states. Did I get your question right?
Yeah, mostly. But let's carry on.
All right. So we proposed this kind of theory then based on these facts, and if you don't want to call it a theory, then I'm also fine with saying that this is a dendritic integration mechanism, but I just want to explain what it kind of does and why it's nice. Somebody has a mic on, so maybe you can turn it off. Thanks. So the first thing is that, if you are familiar with consciousness research, then it's really like separated into people who study the state of consciousness and people who study contents of consciousness. We expand on that on this Frontiers paper. But the point is that most people working on the state of consciousness, they study maybe the brainstem or the non-specific thalamus and how it affects the state of consciousness. Whereas people working on the contents of consciousness, they usually work more on the cortex. And now we here come and say, look, actually, these two are coupled mechanistically because the layer 5 pyramidal cells are involved in both types of processing. So these layer 5 pyramidal cells are the critical nexus for thalamocortical communication. So that's why this kind of research, for my mind, kind of integrates people who study the state and the contents of consciousness. And we also try to explain how this kind of a theory and looking at the thalamus and the pyramidal cells can explain both the state and the contents. Now, the second point, and maybe that's more crucial, is that this mechanism that I have explained to you is highly local, right? It's really, as I have explained it to you, there is a mechanism within single pyramidal cells that can couple and decouple the two compartments of the pyramidal cells. So it seems that, wow, this is way too local. I mean, what can it tell us about consciousness? But of course, one has to keep in mind that there are not one or not six, but millions of these kinds of pyramidal cells in our brain. So fundamentally, when we kind of couple or decouple these neurons locally, then we actually couple or decouple the whole thalamocortical system, as I explained to you already in the previous point. And this is very important. So there has been a lot of research showing that consciousness is related to this distributed processing in the thalamocortical system. But we show that all of this distributed processing can be actually controlled locally. If you sever this connection between the two parts of the pyramidal neuron, you actually also prohibit this kind of thalamocortical activation. Or to put it into other words, I like this, this paper by Athena Demirci and others saying that consciousness rests on the brain's ability to sustain rich brain dynamics. But I would like to add, which depends on this dendritic integration. So you can have this sustained rich brain dynamics only if these two streams within a single pyramidal cell are coupled. And here I try to show it graphically that, in the unconscious state, you might have an activation of the thalamus. This thalamus might even project back to the apical compartments. It might even activate the apical compartments. And indeed, we think that might be crucial for learning, for example. But if this connectivity is blocked from the apical to the basal compartment, then that's it. You cannot have a loop. You cannot enforce this activity. You cannot create sustained, rich brain dynamics. Whereas in the conscious state, when this gate is open and the apical compartment can activate the basal compartment, you can build these recurrent loops. You can sustain activity. You can maintain it, for example, for working memory. But through that mechanism, which is a local mechanism, you can actually control whether the activation evolves and changes and looks like conscious activation, or whether it's very local. And so we actually, in our paper, we also put forward that, we can look at these experiments that have been done, for example, here by Marcello Massimini in 2005. And now we can explain why the TMS in the awake state leads to this activation that is complex and interesting, because this activation can flow and make these loops between the thalamus, between the cortex, with the layer 5 pyramidal cells being the nexus of it. Whereas during non-REM sleep, you can have a local activation, but it doesn't propagate. So this local mechanism can explain the global dynamics. Maybe there's another question somebody wants to ask.
Hi, I have a question. I don't know if my video is up here. So what you seem to describe, from what I can tell, is a difference in neural activity or brain state between when you're asleep and when you're awake, or anesthetized and awake, right? But there are lots of brain differences between when you're asleep and when you're awake. So, like, to call any individual one of them a theory of consciousness, so where does that come from?
Thanks. That's a nice critical question, and a very good introduction into what I will try to explain next. Okay?
Okay.
Yeah. Thanks if you can mute your microphone also. So this is indeed, I mean, if you look into the paper of Mototaka and Matthew, it's a paper of a state of consciousness, right? But I myself have a long background in studying the contents of consciousness. So that was, of course, a key question for me. And, the point we want to make is that that conscious processing, so processing of conscious content, is always affected by prior knowledge. So that this theory, it makes very specific claims about what kind of processing conscious processing is. And coming back to the introduction, when I explained that other theories have to basically make extra assumptions to explain these effects of prior knowledge, etc., then this theory, it has it inherent in it, right? That, during conscious states, this top-down or internal or prior knowledge affects processing. So we have made some pretty strong claims, but we make them on purpose. So, for example, one claim we have made is that, you know, cortical layer 2-3 activities is largely unconscious. And note that this unconscious activity can also be modulated by prior knowledge. So we are not saying that unconscious activity can never be modulated by prior knowledge. What we are saying is that when the layer 5 activation has these two components, the top-down and the bottom-up, then this content is conscious. Whereas when you would have, let me show the image, when you would have simply activation of the basal compartment without the activation of the apical compartment, then this activation would be non-conscious. So this would then account for non-conscious processing within the conscious state. But of course, the question was more like, oh, do you actually have more evidence for this? And let me skip to that. Let me go to this paper. Some of you might have seen it, but this is also a paper coming from the lab of Matthew Lacum, where Naoya Takahashi trained mice to do this classical detection task. That's also often done in consciousness research with humans, where, well, in the sense that there's a very faint stimulus and the animal or human has to detect it. That's the similarity. The difference is that in animals, you do it with a whisker stimulation, which you usually do not do in humans. But he did it very nicely. It's a very complicated experiment where you have the animal gets activation of different, the whisker is activated to a different extent. The animal has to detect it. And then they actually measure the activation of these apical dendrites. And they can show that the activation of these apical dendrites is correlated with the perceptual report of the animals. So when the animal reports that, oh yeah, I perceive it, well, not by saying, oh yeah, but by clicking something or by licking in this case, then there's more activation of these apical compartments. And moreover, what you can do, of course, with optogenetics is you can actually manipulate it. So if you are saying, as we are saying, basically that this activation and manipulation of the apical compartment has an effect on perception, then changing it, controlling it should actually have an effect on perception. And that's exactly what he then showed. Maybe I won't explain all of these plots, but so he did many experiments. So first he had eye clock in these layer 5 pyramidal cells. And here he saw a relatively weak effect, but still that if you inhibit these apical compartments, the detection performance of the animals drops. Then he did several other experiments, for example, by activating the inhibitory SOM cells and by then activating the pyramidal cells, the apical compartments of the pyramidal cells. And what you see in short is that the manipulation of these apical compartments affects the perceptual behavior of the animal. So in the sense that this paper and the subsequent paper that he published in Nature Neuroscience show that, you know, this effect or this theory is not only rooted in one paper, but it's more than that. So we know that also you can actually have an effect on the contents of consciousness by manipulating the apical compartments. So when you upregulate the apical compartments, the animals show increased detection behavior. Yes.
Yeah, but we can also manipulate perception by activating the pyramidal cells themselves in layer 5. You don't need to go to this apical compartment.
Right. But of course, when you have a very specific idea that this context or top down has an effect, and nobody else basically says it, basically others would say, yeah, you can manipulate perception by doing something there in the basal compartment, but these apical compartments I don't care about. And then you show a specific effect of this activation of the apical compartment, then I think you have pretty good experimental data to say that, okay, you're on to something. And remember, guys, when I started to explain my theory, I never said that, that I believe that this is third theory and this is the best theory that there will ever be. And there have been several criticisms that, this is premature, you don't have so much data about it, and so on. But my response is that, sometimes you have to stick the head out and say that, look, here's enough interesting data that could be a theory. And that, again, for me, the main point is to motivate actually other people to come up with much better biological theories, right? So that's my starting point, that I know that the data we have, it's not too much data. It's not so much data as, for example, the global neural workspace theory has. However, some of the predictions we make are much more easier, straightforward to test by a neuroscientist. And that is our point. Okay. Okay, let me skip that example and say that one interesting aspect that I find with these theories that we can now also better explain many types of experience that are purely internally generated, for example, dreams or hallucinations. So I have to stop at some time point, so I won't explain all of that. If you are interested, then please check this other paper we had in 2020. But the main point here is the following. We already discussed this part, right? That we predict that during non-REM sleep, the neurons would be decoupled. And this is one of these testable predictions that the theory makes, and that I hope that soon either we or somebody else will actually test. But there's another prediction that it's harder to do, but still a bit possible, is that, that during REM sleep, that is not exclusively related to dreams, but still largely related to dreams, you should actually have this kind of a coupling. This experiment is very hard to do in mice because mice, if you do experiments on them, it's hard to have them doing REM sleep. At least we in our lab, we saw that it's very difficult. But this would be another prediction that we have put forward, and where we are happy to be wrong, that during REM sleep, you should have this coupling. So when you activate the apical compartment in REM sleep, then this should propagate to the basal compartment. And yeah, so maybe just to kind of show where I'm coming from, I have also done many behavioral experiments. This is one of them. In this experiment, we did this kind of a trick that mostly the subjects, they have to tell us something about the two faces, but occasionally they have to say how well they perceive the square. And then it's a really boring experiment. They do like 100 trials, and then we actually do this. So there's no square. Okay? But we ask them, nevertheless, okay, well, how well did you actually perceive the square? And, we would expect that everybody answers, oh, yeah, I didn't experience any square. but we have done it in 3 different experiments, actually 4 different experiments, and saw that actually many participants report that they have a clear perception of a stimulus that is not there. So this is one example of how you can have something like a hallucination in normal people. And I mean, other labs have also shown effects like that. But this is just to show, again, I mean, this was kind of my background, and this is why this kind of a theory that explains how this might happen is very exciting for me, because now we can go back to this result I showed from Naoya Takahashi and others, where they did it with mice, and they showed that, you know, look here, when there's actually no stimulus, there's nothing, there's no activation of the basal compartment. But if you activate the apical compartment, then the mice report that there was a stimulus. So there's none, as in our human experiments, but because the apical compartment is activated, the mice report that there's a stimulus. So our point is that this kind of a theory explains why in certain situations, expectations, predictions can generate these kinds of hallucinations, even in healthy subjects. Okay, and let me end by saying that, of course, I have kind of explained to you our theory and said that, you know, I would like to see more theories like that, but fundamentally we agree that this is compatible with other big theories. As said, these big theories currently rely on something like the idea that these are point neurons. They don't really care about the complexity of the pyramidal cells, but you could do that. You could actually say that each of these units in these theories has these 2 compartments, and you can basically cut off the communication between them. And the way we see it, it would actually benefit both the integrated information theory and the global neural workspace theory, because then they would have a very specific cellular mechanism that would explain certain properties that they have. So fundamentally, my point is that these are super exciting times in neuroscience and in consciousness research. And I urge really everyone to think about what would a good theory of consciousness look like. And, you know, let's not be constrained by old theories. Let's not be constrained by this theory I have proposed today, but let's just agree that we know now more about consciousness than 20 years ago. Thank you very much. I'm happy to take questions. And again, it has been a pleasure to be here.
Thank you very much, Jaan. There are a few questions in the chat. First, Jian Shin said that in humans, layer 2-3 neurons also show dendritic spikes and compartmentalization. So do you think that in human cellular layers integrating top-down and bottom-up information are expanded both to layer 2-3 and layer 5?
Hi, Jian. The right answer to say at this point is that we don't know, right? We know in mice indeed and rats that, you know, layer 5 cells have this difference to layer 2-3 cells, and what we can say currently is that our theory is based on layer 5 cells and their properties. However, as we have learned indeed over the last years, is that in humans you might have layer 2-3 cells that are not really similar to rodent layer 2-3 cells, that look like something new and that do exciting things. I mean, people in Matthew's lab have also shown that. So fundamentally, I think we are not against saying that or agreeing that layer 2-3 cells do something similar. Simply, again, by making strong claims like saying that layer 2-3 activity is not conscious, we hope to provoke research. We hope to provoke researchers to show, oh, you're wrong, actually. And I'm always happy to be wrong.
Okay, and another question by Regila D. What does DIT add to already suggested theories of consciousness by properties of predictive processing, for example, by Nielsen and Friston? And does DIT have has a computational aspect?
Yeah, thanks for the question. And so predictive coding, you could again say is a very based on this, this cascade of predictions and prediction errors. But of course, we would say that our theory is much more neurobiological. So predictive coding makes many claims that that are not so neurobiological. In the end, it is a computational model. So our theory is more neurobiological. Theirs is more computational, but fundamentally they can be kind of integrated. So predictive coding theories can take this mechanism as something crucial for how the predictions are computed and how this is done in the brain. And we, on the other hand, can take at least some of the computational aspects of their theory to build our theory. So we haven't done that at this point, but fundamentally, when I have also spoken with Anil Seth, we feel that this is compatible.
Okay, and Hadar Levi Aroni asked, what would be your prediction for EEG and fMRI measurements, if there are any, and that you had previous work on the P300 marker wave, and as a consciousness marker for as a marker for consciousness, and what is the expected latency in this case? It would be the P300 or earlier?
A great question. Yeah. So I worked previously, as you have seen, I have worked on many different things, and I have worked also on EEG, and we studied the neuromarkers of consciousness, and we studied the P300, which is a component of the EEG response, right? And this is indeed something that is a pressing issue for us to understand. Can you actually, when you work with humans, can you actually study this theory, or it makes no sense for you? So on the one hand, we are now looking into this very good data that has been collected from the layer-specific fMRI. There are some data sets out there with layer-specific fMRI where you can distinguish the activity of different layers in the human brain. And we want to see whether we can actually kind of model that activity, so make specific predictions about the fMRI time courses, and then actually do an experiment with this layer-specific fMRI. And then the same question is about EEG. We have some suspicion that indeed this kind of P300, as you see, a really big event in the electrical activity of the brain, is associated to a large activation of these layer 5 pyramidal cells and their coupled activation. So this was shown in a previous work by Mototaka and Matthew. I mean, they showed evidence for it, that this is possible. But fundamentally, some of you who might be working on EEG also know that there's this negative deflection, sometimes called visual awareness negativity, or now even perceptual awareness negativity. And we think, and based on the dipole modeling, we think that when you activate the apical compartments, then this is the negativity, because it makes sense based on the dipoles. If you activate the apical compartments, you should get a surface negativity. So if you activate apical negativity, but if you get into this large mode of propagation and a lot of layer 5 cells firing, then you get a P300 as we see usually in the experiment studying consciousness.
Thank you, and we have time for one more question. So Tomer Bach asks, is the somatic compartment still being activated by feed-forward signals during anesthesia?
Yes, exactly. I mean, this is has been known for a long time that, under anesthesia or also in non-REM sleep, you still can activate the cell bodies of the neurons. And so the feed-forward signals activate the basal compartments, but as they are decoupled from the apical compartment, then you can have some activation. They can even send some activation to the thalamus, but it doesn't do anything. So it stays local. It has no consequence, right? So yeah, that's the answer.
Okay, thank you very much, Jaan. So everyone, next week we do not have a seminar on Thursday because we have the conference of Virtual Glimpse into the Future during the week, and we'll meet again in 2 weeks for another Zoom talk by Ken Norman. So thank you everyone for joining.
Thank you, guys. Thank you for the great questions. It was an honor to be here. All the best.
Thank you, Jaan, and we'll see you in the meeting soon.
Yeah, ciao.
Ciao.